I'm speaking to you today under two roles. First, I am substituting for the keynote speakers, the CEO and president of my company, as well as the head of the Drug Discovery Institute in Duke University, Professor Organisers, and also representing our paper in EPMA journal on the development of a marker for Alzheimer's disease prognosis and its utility in clinical trials. And I believe this is a pioneering example for the PPPM field, which I think could be also applicable to other fields, disease areas, and I hope you will agree with me. So let's, without further ado, move on. So just kind of take a step back on the general status of the development of drugs and how, and you can click through this one. We've been pretty successful in the development of drugs so far in the last several decades. So in general, there are good treatments out there, and there has been the regulators have been working with the companies and generating good drugs. However, as time progresses, some drugs have been removed from the market. There is decreased productivity. We're having a hard time finding new effective treatments and new pathways that will outperform the drugs we have. And so we're at a crossroad in the industry of how to progress and how to better the healthcare. And it is believed that PPPM will be the solution to find a way forward. And it has been recognized by payers as well as their regulatory agencies that this is truly the way forward. So it has become through the last decade from just talk to actual actions. And you can see that some pharmacy benefit managers are actually running studies to show that there is economical benefit to using markers and biomarkers. For example, the warfarin example and genetic markers for that. Medco has shown that there is true value, true benefit in reducing hospitalizations, and of course, go on by that. And these are just a couple of new guidances. This is just from the last several months. Since February, the FDA has generated 5 different guidelines in this area about the use of pharmacogenetics in clinical development, about the EMA followed by their view as well. Next. The use of companion diagnostic, how to generate that in time so that the patient can use the drug and the diagnostic in a timely fashion, and so on and so forth. So, and these are just 2 examples from the last month, from August, of 2 drugs with companion diagnostic that have been released to the market. So this is truly the time when we see translational medicine. And again, I'll just go through this quickly for the interest of time, but really, as we all talked about in the last several days, validation and appropriate development is really important. So there is a phase for hypothesis generation. We do that in early phase clinical trials. Then in the next step, we do testing and confirmation of these findings. So that usually is in phase 2 or in similar fashion. And then we go into phase 3 and we do the validation of the marker. And that is currently the way things are done in major or in the big pharma, not only for genetics, but also for genomics and other biomarkers. And it's truly the paradigm that is prevailing. And of course, as we go through that process, we start with many markers, whole genome, genomics, whatever it is, and we end up with those few pinpointing biomarkers or this signature that is very specific, needs to show specificity, sensitivity, positive and negative predictive value, and so on. We can go through this one. And these are just a couple of examples, and I'm just going to concentrate on the very last one in red and how we develop the COM40 marker, and how are we going about validating and using it in a clinical trial for the prevention of Alzheimer's disease. So here we can see the very first discovery of the ApoE marker, the ApoE4, and its association with Alzheimer's disease. Professor Alan Moses and his group made the initial discovery, and it was since confirmed and replicated many times as actually the single most significant risk factor for AD. However, it is not recommended in clinical use. It is not, the predictive value is not strong enough, and it's also not very prevalent, and therefore it is not part of clinical practice. Also, the most prevalent allele, so the ApoE gene has 3 alleles, the ApoE2, 3, and 4, and the ApoE3 has been, which is the most prevalent one, has generally been described as benign or neutral. But our discovery has shown that this is actually not true, and the ApoE3 allele itself is, in combination with a different allele, can be subdivided into 2 groups. Some of them are at high risk, and some